Most people learn that infections come from living microbes or from viruses carrying genetic material. Prions break that rule completely. A prion has no DNA and no RNA. It is not a cell and not a virus. It is a protein that has adopted the wrong three-dimensional shape, and that shape itself is the threat. The danger is not in a genetic code but in a structural template. When this malformed protein contacts a normal version of the same protein, it can force that normal one to refold into the same harmful form. This creates a biological domino effect. One bad fold can become many, then thousands, then millions over time. That is why prions are so unsettling in medicine. They show that information in biology is not only stored in nucleic acids. Under certain conditions, information can also be stored in shape. In prion disease, shape becomes contagious. To understand the danger, begin with the normal prion protein, often called PRP. This protein is naturally present in mammals, especially in nervous tissue. Scientists still debate its full day-to-day role, but the key point is that the healthy form is part of normal biology. Disease starts when some of these proteins misfold. That first misfold can happen in different ways. It may occur sporadically, meaning by chance during normal protein turnover. It may be driven by inherited mutations that make misfolding more likely. Or it may follow exposure to an already misfolded prion protein from outside the body. Regardless of origin, once the abnormal form appears, the process can amplify. This is why prion diseases are grouped into sporadic, genetic, and acquired forms. Different starting points, same catastrophic endpoint, progressive damage to neural tissue. The conversion process is the core mechanism. Proteins are not rigid stones, they are dynamic molecules whose function depends on folding. In prion disorders, the abnormal form acts like a molecular mold. When it binds the normal form, it lowers the energy barrier for misfolding and nudges the normal protein into the same pathogenic conformation. Newly converted molecules then repeat the process. Over time, this creates self-propagating assemblies. Many of these structures organize into fibrils and larger aggregates known as amyloids. At first, this may sound slow and microscopic, and it is. But neurodegeneration unfolds over years. A tiny seeding event can eventually produce widespread pathology. This is one reason symptoms often appear late, after substantial invisible damage has already accumulated in the brain. Prion diseases are fatal neurodegenerative disorders because the brain is especially vulnerable to this kind of protein corruption. Neural circuits depend on precise cellular architecture, stable signaling, and long-lived cells that are hard to replace. As misfolded proteins accumulate, cells experience stress in multiple systems: protein quality control, membrane integrity, synaptic signaling, and energy balance. Aggregates can disrupt normal trafficking and overwhelm clearance pathways. Eventually, neurons malfunction and die. Clinically, this appears as rapidly progressive decline in cognition, coordination, behavior, and motor control, depending on the disease subtype and brain regions affected. The progression is relentless. Unlike many infections that can be cleared and many chronic diseases that can be stabilized, established prion disease currently has no effective curative treatment and ends in death. The disease list across species is important because it reveals both biological consistency and public health risk. In sheep and goats, scrapie has been recognized for centuries. In cattle, bovine spongiform encephalopathy, or mad cow disease, became globally known after feed practices enabled amplification and spread. In deer and related species, chronic wasting disease continues to raise concern due to persistence in animal populations and environmental contamination questions. In humans, Creutzfeldt-Jakob disease is the best known, with sporadic forms most common, plus inherited and acquired. Variants. Across these conditions, one theme repeats: different hosts, different epidemiology, but a shared mechanism of protein misfolding, aggregation, and neurodegeneration. This cross-species pattern strongly supports the prion model as a unifying explanation rather than isolated medical mysteries. A key historical breakthrough was naming prion as pernicious infectious particles. That label was controversial at first because it challenged the central intuition that infection requires nucleic acid replication. But experimental evidence kept pointing back to protein conformations as the transmissible unit. This shifted biology in two major ways. First, it expanded the concept of heredity-like information transfer to include structural templating. Second, it forced infection control and pathology to consider a non-traditional agent with unusual resistance properties. The prion framework did not erase genetics; inherited mutations still matter. Instead, it connected genetics, protein chemistry, and transmission into one model. You can think of prion disease as a convergence problem: sequence sets vulnerability, structure triggers propagation, and tissue environment determines where damage accelerates fastest. Why are prions so hard to contain? Their structural stability is a major reason. Many infectious agents are inactivated by standard sterilization or disinfectant routines designed around cells, membranes, or nucleic acids. Misfolded prion proteins can remain stubbornly resistant to conditions that would neutralize many pathogens. This creates special concern in healthcare settings, where instruments contacting high-risk tissues must follow stricter decontamination protocols. The issue is not routine casual contact but specific exposure pathways combined with inadequate processing. Historically, concern about iatrogenic spread, meaning transmission related to medical procedures, pushed major upgrades in sterilization guidance and risk management. In practical terms, prion force institutions to treat protein contamination as a high-level biosafety challenge, not just a routine cleaning problem. Another crucial concept is that prion are not simply static clumps. The proteins involved can exist in multiple conformational states, and some pathogenic states can template their own structure with remarkable fidelity. This helps explain strain-like behavior, where distinct disease patterns emerge despite involving the same underlying protein family. Different conformers may favor different brain regions, progression speeds, or clinical features. That complexity complicates diagnostics and therapy design. A treatment that destabilizes one pathogenic conformation may be less effective against another. It also means researchers must study not just whether a protein is misfolded, but exactly how it is folded in disease-relevant contexts. Structural biology, biophysics, and neuropathology all become essential pieces of the same puzzle. Prion science also influenced how we think about other neurodegenerative diseases. Conditions like Alzheimer's, Parkinson's, and ALS are not classified as classic infectious prion diseases in the same way, but many show prion-like mechanisms. Misfolded proteins concede further misfolding and spread pathology through neural networks over time. For example, abnormal aggregates of proteins such as alpha-synuclein or tau may propagate in a templated manner within the nervous system. This does not mean these diseases are contagious in ordinary social settings. It means the internal disease logic, at the cellular level, can mirror aspects of prion propagation. That insight has reshaped research strategies, pushing scientists to target early seeding, intercellular spread, and aggregate clearance before irreversible circuit damage accumulates. Multiple system atrophy, linked to prion-form behavior of alpha-synuclein, illustrates how the field keeps evolving. For years, PRP-centered diseases defined the prion category in mammals. Then evidence showed that another protein could display key prion properties in disease context. This broadened scientific discussion. Perhaps prion biology is less about one specific molecule and more about a general principle of pathogenic conformational templating. If true, the implications are enormous. It could unify mechanisms across several disorders now treated as separate categories and help explain why neurodegenerative diseases often progress despite different triggers. At the same time, caution is important. Expanding the prion concept should remain evidence-driven, with clear distinctions between classical transmissible prion diseases and disorders that are merely prion-like in propagation mechanics. So where does treatment stand? The challenge is timing and target accessibility. By symptom onset, the brain may already carry a heavy burden of misfolded protein and cell loss. Therapies must either prevent initial misfolding, block templated conversion, enhance clearance of toxic species, protect neurons from downstream stress, or ideally combine these approaches. Each step is difficult. Blocking conversion requires high specificity to avoid disrupting normal protein function. Boosting clearance risks inflammatory side effects. Delivering drugs across the blood-brain barrier remains a major obstacle. Even diagnostics are hard because early disease can be silent while pathology expands. Progress is real, especially in sensitive detection methods and mechanistic understanding, but translating that progress into robust clinical outcomes remains one of the hardest problems in modern neurology. The biggest takeaway is simple but profound: biology can fail through structure alone. Prions teach us that a protein's shape can carry pathological information, self-amplify, and devastate the brain without any viral genome involved. They also reveal a larger pattern in neurodegeneration, where misfolded proteins may behave less like random debris and more like active propagators of disease state. That shifts how we think about prevention, diagnosis, and treatment. Earlier detection, confirmation-specific targeting, and strict control of high-risk exposures become central priorities. Prions are rare compared with many disorders, but scientifically they are foundational. They force medicine to confront an uncomfortable truth: in the nervous system, a single molecular misfold can become a long, silent cascade that ends in irreversible loss unless interrupted early. If you found this helpful, please like this video and subscribe for more explainers.